very special guest, a returnee, if you may, Dr. Badlal McCreath. She's onto some stuff that is out of this world that we take for granted. The last time we met Dr. Simone, we spoke to cells and the need to deal with cells that will match and best fit with persons of African descent. And you're talking about prostate cancer. Speak to us as if we have never heard anything about it before, and where are you with it now? So give us that overview, review, if you may, of the situation.
So you know that the building block of life, be it animals or plants, comprise of cells. And if we can access cells in a way that we can grow them outside of the host origin, we can study them. We can study different diseases and we can better be able to treat, manage, cure. So for centuries, for a very long time, scientists attempted to grow human cells outside of the body in a prolonged and sustainable way for that simple reason that they could understand diseases. So I believe the first recorded attempt at this was in the 1880s, and the first successfully developed human cell line didn't happen until in the 1951 from a woman called Henrietta Lacks. That show, I mean, her story was showcased on HBO and Oprah Winfrey starred in it, just to point or highlight the significance of such a feat. So as scientists, what we do is we access these cells from overseas suppliers and we grow them in our labs for the same reason. Whatever disease you're studying, cancer, whatever disease, you grow these cells, you formulate your protocols and you develop those along that line. So just to backdrop, for my PhD research, which I had discussed with you on the first interview, but just to recap, I was able to look at or grow several cells that we purchased from overseas. And we screened these cells, or no, we screened the Jamaican natural products, against these cell lines for the sole purpose of identifying natural products that could reduce the viability of these cells. And so we could deem them potential chemotherapeutic agents. And what we did as well, we compared the effects of our natural products with known drugs on the market. So the cells that we purchased were breast, colon, and liver. And we also purchased normal cells. And what we found from our data was that several of our Jamaican natural products were more effective in killing the cancer cells than known drugs on the market. And to add further to that, some of our natural products did not harm the normal cells compared to the known drugs. So that created a buzz. Several awards were received, and we received grant funding because of the significance of that data. And so I was on the verge, finishing the PhD and figuring out what's the next move as a young academic. And one of the things that I was thinking of was to test those natural products in animal models. So there's a system to developing drugs. The first is you test it in the cells, and then you select or you identify the promising ones, and then you take those to animal models, and then you select the promising ones from that, and then you take it to human beings. So that's the systematic step. So I had my eyes set on the animal models route, but what it caused me to do was to investigate my data a bit more. And I realized all the cell lines that I had used in my project were Caucasian cell lines. Mm. So this started a buzz of questions, inquisition on my part. Why is it that these popularly used cells are Caucasian cells? And if it is we want to develop our Jamaican natural products as anti-cancer drugs, then is it reasonable that we test them on Caucasian cells to identify treatments to treat Black people. That's just common sense right there. So I checked and I realized one, there were no cell lines developed within the Caribbean. So no cell line was available that was of Caribbean ancestry or descent. When I further probed, I realized a grave disparity in the ethnically available cell lines, so Caucasian compared to Blacks. So I narrowed my search to the two prominent cancers of concern for us as Jamaicans, Caribbeans, Black people, and those are prostate and breast. So that led me, the prostate route, Michael, 98% of prostate cancer cell lines are Caucasian in origin. And the one, of the few Black ones available, they recently pulled one from the market because they found that it's not Black and it's not prostate. So that was a great reason for me to say, hey, if it is that I want to develop drugs that can treat cancers within or among Jamaicans, Caribbeans, Black people, then it only makes sense that we have tools that represent our people.
Yes.
And so I took on this ambitious notion of developing cell lines, which would have been the first from the Caribbean. Along the way, several persons advised me, several senior professors overseas advised me that developing a cell line is so technical, it is so um, risky. There's a less than 10% success rate in doing it. So even though it's been around for so long, you find that the library or catalog is relatively low. And that's because it's such a difficult thing to do. So some of them said to me, Simone, I will help you with other things, but I won't help you with this because I've tried it and it didn't work. There was one that I reached out to who had done it, or he said he had done it, and he said, I would advise you not to do it. And it took time. And I had a very or have a studious PhD student who worked closely with me. So this was a team effort. This wasn't just a Simone effort. Research is never about a one person thing. And we have urologists on board who supply us with the samples. It's been what, 5 years now. Some persons along the way said, Simone, if I were you, I would go another route. And when I met with you last, I said to you, Michael, we're almost there. And you said, Simone, as soon as you've done it, come back. So we actually did it in December of last year into January of this year. But we have been hush hush because we're trying to get it published. And we also wanted to test it, prove it, certify it before saying anything. So I'm happy to.
Help me know so that when you say you did it, put in clear and precise terms what exactly that was done. I get the sense of it, but I want to be sure that all the viewers are able to say what it is that your team was able to accomplish uh, late last year and early this year.
So we were able to develop the first prostate cancer cell line from the Caribbean. And it so happens to be a Jamaican prostate cancer cell line. And we were able to refine or develop a protocol that is unique to this. And so when we publish it, this will be a novel area to science in addition to what they currently have.
Let me tell you what my understanding of science is. My understanding of science is that when you repeat it, you get the same result. Yes. So what are the stages you took? How do you get a cell line? Do you take somebody's cancered prostate and put how does
Very,
it
very
work?
good
Break it
question.
down for me so I can get the sense that as a carpenter I can build one.
So as I say, we work with the surgeons, specifically urologists. And when they are having a procedure going on, be it a biopsy or a surgery, they get in touch with us. So we go to the hospital with our different kits on hand. We wait to collect that sample. We also carry out, well, before we collect it, we have to get consent from the patient. So we inform the patient of the research and ask if they would be willing to participate or donate their sample to us. we protect persons' confidence and their trust. So we go through tried and tested ethical documents to ensure that everything is on point. Then we take that sample immediately to the lab and we process it. So you cut it, you do a lot of things, but you probably don't need to go into all of that. You expose it to different reagents and materials. So um, each human being, each cell, each lab has different conditions. And so one protocol does not work for all. And so even though we have defined or created a protocol for this cell line, there's no guarantee that when we use the same protocol, it will work for another one. So we have to be well aware of the different dimensions and angles, what to tweak, how to tweak. And the experience that we have garnered so far, I think, positions us well to create or mass produce other cell lines from other African, Jamaican men. One of the tricky parts about it is that the cell line is classified as cell line once the cells are immortal. So you need the cells to not die. And that is
Ooh.
one of the challenging steps. Another challenging step is the cell has to be free from contamination. So it has to be pure. So bacterial contamination is highly likely, especially since when we are developing the cell line, we have to not expose the cells to antibiotics because you don't want them to become used to it. This
Yes.
is a tool that we're gonna sell to other suppliers, sell to, we're going to make, we'll donate it to other labs. So you have to ensure you're not giving them a antibiotic resistant cell line. So, I mean, the dimensions and the complexities exist with creating this tool, and it took us a couple of years.
Yes. So
Um,
let me again drill down for those who might just be joining, and that it's about a cell line that will more akin and in on the same page, if you may, with those of African descent for Black people. What's the value as it relates to having the cell line? I come in, God forbid, I have prostate cancer. How does your cell line help me? I want to drill it down to the individual. So those who might think, oh, a doctor thing that, a research thing that, will see the benefit of what you and your team are up to.
so what we recognize is there is a disparity in prostate cancer occurrence among Black men and white men. There's a disparity in how Black men with prostate cancer respond to chemotherapy drugs compared to a white man with the same. So what we find is Black men are 2.5 times more likely to die from prostate cancer than white men. They are way more likely to develop the disease than a white man. So if you have two men, Black and white, with the same grade type of prostate cancer and you give them the same drug, research shows that the white man will respond more favorable to that drug than the Black man. And so the white man would experience better quality of life, and I guess better longevity of life, if you will. So with that disparity, you have to then take a step back and say why. Why is it that after science has been around so long and with us evolving so quickly, that there is this disparity? And when you look at it, the tools that we use to study the disease are primarily from white men. When you go to, so at the basic science level, which is the cellular level, as I said earlier, 98% of these are from white men. When you take those and you go to the animal models, you're using those same cells. Then you go to clinical trials. Most of the men enrolled in clinical trials, more than 90% of them are white men. So if we are studying the disease in terms of understanding prostate cancer, with the goal of developing treatments or cures using primarily samples from white men and white men, then that, in my opinion,
Yes.
explains why they get greater benefit from the treatments that are currently developed.
It sounds to me as if the whole approach to medical science is also systemic in terms of persons who of property and money and possibilities that there is, and therefore the the study resembles the world in terms of you know, equity and non-equity, if you may, and somehow you have come in not only to do this but to champion the cause of persons of African descent.
The story of Floyd Green apparently permeates science too.
Oh, indeed. That's a good shot. That's a good shot. But here's what where I want to take this now. You are going to, you say this has worked, and you are going to take it to publish. Scientifically, what's the significance of having it published scientifically? Where do we go from here before I get on to the broader things of you call the Jamaican natural products? Where do you go in terms of publishing?
So science is validated through publications or journals. And what you have are a group of experts that sit on different um, bodies that vet the work that people are conducting. So even with COVID now, the science has to be rigorous. So it can't be Simone is in the lab or Michael is in the lab. They come up with something and it's secure. We have to vet what you have done, why you have done it, and the quality of the data that you have produced. That is, so it's a very rigorous system. So a scientist progresses in his or her career based on the number of publications that they obtain and based on the quality of publications. Because with anything in life, you're going to have good and bad. So you do have publications that are not vetted rigorously, while you do have those that are on the hard side, you know. So the challenging side to get in. So for us, as much as we've said we've developed this tool, we want to ensure that the science that we have utilized to develop this tool is vetted by the scientific community. And we also want to ensure that we state that this is what we have done. So it's, I guess, a substitute to a patent. So far, we have consulted the different journals that are specialized in prostate cancer. We've looked at them. And for me personally, I'm interested in going into a journal that exercises strong rigor. And I've already contacted one of the editors for one of the best prostate cancer journals, and they have agreed to accept to review the manuscript. And so they're now waiting on us to submit it to them. So the last set of data that we were waiting on, I actually just got an email 2 days ago to say that that data is ready. So I'm pushing to have the paper wrapped up within a week, 2 weeks max, so I can submit it to that journal and hopefully have that manuscript published before the year is over.
Um, Let me ask you, does this make you nervous? Because you have, for example, one, taken on an area where most people have not gone. You know, 98%, you know, cell lines for white people. You persons have looked at you and said, well, maybe you shouldn't bother with this, you know, I'll help you with something else if you're interested in. Now you're opening up yourself. You have gone the scientific route before. You're now saying to them, let's us hear from you. But I suspect those very journals might be made up of persons not coming from the parts of the world that you came from, and would still have that inherent bias. Help me, is there the politics of it? I'm trying to see where your headspace is when you approach this, and then you get wider out. Now, here is my work. I believe it can stand the scientific test of time.
So the editors or those who are in charge of the journals, they usually work with reviewers. So there is a system in place to um, prevent or minimize biases. So they'll ask you, do you have reviewers that you would suggest to review this manuscript? And you could suggest persons who are experts in the field. They could also say, is there anyone that you don't want us to send? Because you could have an interaction with a reviewer who you think might um, display negativity unnecessarily towards your work, and you can say, okay, could you please not um, utilize that person? So what happens is they send the manuscript out to several different reviewers across the world in as much as an unbiased way as they can.
Mhm.
And then these reviewers either say to the editor, we agree that this manuscript should be published, it can be published as is, or this manuscript has to address the following areas before it can be published. And then the editor will send you either we will accept it or no. The editor has that power or authority, or they can say, hey, these are the comments that the reviewers have suggested. Could you please look at them, address them, and send us back a reply or a response to these? So it's not so much easy to create or involve that in there. It's pretty much, in my opinion, kind of, I guess, fair from my experiences so far. But nothing is perfect, right?
Yes, yes.
Um, yeah.
The rigor. It sounds rigorous to me. You can send your manuscript. And by the way, how long is a manuscript? What's the size? I'm getting the sense now is reams and reams and reams, a big folder as opposed to a small folder. But so to address that in terms of the size of it, what's in it, and the to and froing. So the editor may very well say, "You have come back with this, but yet we still want to do this." So
Yes.
it is
Mhm.
a paper that can keep you can keep working on. So address the issue of the size for me and the rigor.
So you have different types of manuscripts. You can have a short article. Um, you can have a review paper. So that's a manuscript that you review the existing information that's out there. And then you can have a manuscript that expresses or outlines novel research, which is what we're doing. And that can range based on the journal we're going into, maybe 10 to 15 pages, maybe a little more. And what it does is it has what you're doing, the aim of your research, why you're doing it, how you have done it, what did you get, and what's the implication of what you got. And I suppose where you would, or what's the next step for you.
So it's you don't give up your scientific thing and the little square thing and the root cause and none of those things that you make it readable, understandable. Help me, you're talking to scientists and it's for
Don't-
scientists.
This
What's the detail like? I just want to get a sense of your pain.
is very detailed. So someone who is not in science or has a scientific background, they would not find it easy or readable. So it would be off-putting for someone without a science background because it's not sugarcoating or simplifying. It's deep science. So yes, if you need to talk about the deltas, the gammas, you put all of that.
Okay, I figured the deltas and the gammas would have gotten in there, but here's what I want. I'm interested now. So you have the cell line, and let's assume that the journal say, well, and even after two and three runs, and say, well, explain this further because you didn't have enough delta and you didn't have much gamma in this part. So help us. And the rigor comes through. Where do you go after that? How do you interact? So you've built a cell line. Don't you need several cell lines? I know you say they live forever. I mean, wow, how to be a cell and live forever. It's the only thing I know that is everlasting. Yes. What do you do? Where does the story go from there?
Okay, so to answer the first question, if the editor comes to you and say, hey, this needs to be done, you can do either one of two things. You can um, discontinue attempting to publish in that specific journal and go to another journal.
Ooh.
Or you can go back to the lab or if it is that they require you to just do a bit more literature search and furnish that manuscript with what they're suggesting, because it's highly likely that whatever they're putting forward strengthens the science of the manuscript. So it as an author, you want to take heed to the advice that the editor slash reviewers provide. Where do we go from now? We go to um, expanding what we've done. So another awesome thing that happened for us is we were able to secure funding from the United States to continue this research. So I submitted an application alongside my mentor, Dr. Camille Reagan, and several other mentors, Professor Wayne McLaughlin, William Aiken, and overseas mentors who guided me to put this application together. And it was rejected at first because I believe we submitted it in 2018 first. They rejected it, so we resubmitted it in 2019. And each time it took me months to work on it because
Yes.
the application is rigorous. It's hundreds of pages. It's so many
Ooh.
different documents. It's like you're doing a PhD thesis all over again. And I had a very good team, an exceptional team, and we sat down, we reworked the application, and we put forward that we need further training, and we put forward that we need to develop additional prostate cancer cell lines. It is extremely difficult. I do believe that I may be the first person to secure direct funding from NIH from UA, because you do have persons there to secure funding, but it's usually done alongside a US entity. So I believe so. The university had to go through a lot of processes, setting up systems to download or access these funds, and it's for over 5 years, and it's around $500,000 to prolong, make more cell lines, because they do realize, the scientific community realize that we have to address this issue. It's significant. It needs addressing.
It's another case of Black Lives Matters.
Yes. Black cells matter.
Yes, yes, indeed. Here's where I want to dovetail this now, and I'm sure you will dodge behind your findings and your science, but I'm going to test you anyway. You made the point earlier that, well, is this a natural path? Is this where does it stack up? Because you're saying that Jamaica's natural products will kill some of these cancer cells and not destroy as many as the good cells. Where did this come from? How did you? Because when we think of science and tablets, we think, we take out the prescription and we do it. But the, for me, a natural product is in its natural form. So, how much can you tell us about that without giving away your life secret?
When I say natural product, for that particular research, and let me also mention that that was done under the supervision of Professor Rupika Delgado. What we did at the time was we worked in conjunction with the Department of Chemistry at the University of West Indies. So the Department of Chemistry, one of their primary goals is to identify novel agents, be it from the marine agents or from terrestrial plants, whatever. That's their goal. Another goal is to synthesize novel agents. So what they found was they would have a stack of compounds, some of them natural, some of them synthetic, without any form of biological activity data on them. So we would access these compounds from them, devise the protocols, and test the compounds against or within the cells that we purchased from overseas. And because this is science, if you're going to say our natural compound that was isolated from that soursop tree, has anti-cancer properties. On what merit are you stating that? So we would have to test it against a drug that is deemed anti-cancerous. So, for example, a drug called doxorubicin or tamoxifen or fluorouracil. So we tested those drugs against the respective cells. So if fluorouracil is known to treat colon cancer, we would test it against colon cancer, and we would compare the effects this specific known drug is having against the colon cancer cells to our natural product. So say this drug is killing the cells at a degree of 16 micromolar, and we would call that value IC50 value. Then we would say, is our natural product giving a lower value or a higher value? If it's giving a lower value, it means that less of our product is required to cause the same effect. So our product is more potent. If the value is higher, then it means more of our product gives that effect. So that's not a product that you would want to further investigate. You want to hone in on those that are either comparable to the known drugs or more potent. And then further, you want to test those or honing on those that aren't harming the normal cells. Because ultimately, one of the grave side effects of anti-cancer drugs is that they kill normal cells. And so you go through these, I mean, the vomiting, the hair loss, all of that. So we want to develop drugs that people can take with very little to no side effects.
You when you say natural still, and you use soursop, for example, I'm sure it's just by the way, um, the the sense that some people out there when they're coming to the last and have their cancer, they everybody tell them about these bushes. Everybody tell them about that. Is there any scientific evidence that some of these things in its natural form and not in the compound, whatever you have in the lab, can and does assist in any real way? Or is it just a mind over matter?
Is it anecdotal? um, there is science. Um, there is science there to prove it, and I believe if a patient tells you that they were at their last and the only thing that they changed was that they started to drink a cup of soursop juice every morning, and they noticed after 2 weeks that lump that was in the stomach or breast is gone, then you have to pause. You have to sit down and think, okay, if the only thing that they changed was that they discontinued cancer drugs, you have to sit and pause. And what science does, though, is we try to identify the individual ingredients within the teas that are responsible for the effect, because we can we can mass produce those individual ingredients and sell them as drugs. And that doesn't necessarily mean that individual ingredients are the best route. Of recent, we find that different ingredients in a tablet may be more ideal when you have the synergistic effect of different things coming together as opposed to one ingredient. We might need multiple pathways within the body to be triggered, activated, or downregulated to get the ideal effect. And one ingredient may not do that. So we are expanding science in a way where we're not just focusing on one thing anymore, but focusing on the different things that affect the different pathways
And the
and seeing if we
combination
can.
thereof. Yes.
Yes.
Um, as you move forward now, you build out the cell line and you go in and you take the remains of the biopsy and you take some of the cancer and you go in your lab and you break it out and then you immortalize these things, make them live forever. Uh, aren't you going to be needing more patience and consent and what's the ratio one human being to a cell line? Is it do for I'm measuring, I want building blocks. I want to hear how this thing work.
Well, for this cell line, it took us this the 28th sample to get it. So we've named this sample ACRJ PC28. ACRJ because that's the name of the lab, Anti-Cancer Research Jamaica PC because it's prostate cancer, and 28 because it's the 28th sample. So it took us 28 samples to get this one right. So 1 divided by 28, what's the percentage rate for that?
You tell me, you do the math. The other issue here is while it was 28 for this one, it may be 30 for the next one, or would it fluctuate, or do you find it's the
It's
general?
highly unlikely that it will be 38 because there are different steps in the protocol for developing the cell line. And the first 2, 3 steps, um, we've managed to optimize the protocol to a point that we've gotten 100% success rate. The final step of the protocol was the immortalization aspect that was giving us some issue, and we were able to do it, but we can't say that we have 100% success rate because this cell line was the first one to accept that. So we've so for the other samples, we know that steps 1 to 3 will be good. So once we apply the immortalization step and it takes it, we shouldn't be having that many challenges with subsequent samples. We don't anticipate that.
It's the subsequent samples that I'm after. That is it an ongoing thing for life, for the lab, that you will need and continue to need
New
patients and their
samples, yes,
consent,
yes.
and you know, to what extent then is there any overseas involvement, not by way of research, but by building the cell line? Do we have to send anything away in our frozen box to come back to us and what have you, or do we get it all localized here?
You know, Michael, for this particular cell line, I am happy and I'm proud to say we did it all here. However, we did go overseas for training, both myself and Henkel, my PhD student. And for a few samples, Henkel did take with him overseas in hopes of producing the cell line there, and it did not work overseas. So he came back. We brought back the expertise that we got there, and we developed it here. What we did do, however, was after we developed the cell line, we shipped a sample overseas because we wanted to verify the authenticity of the cell line. So we wanted to verify, is it novel? Is it human? Is it free from bacterial contamination, mycoplasma? All those checks and balances needed to be done before we come to say, hey, we've developed a cell line. So Henkel and myself waited patiently for that report when it came back, just like how a student waits for their C-sec or your final exams at UA. And I remember the report came to me directly, and I didn't tell him over the phone. I actually took him out to lunch and told him to his face, and he just broke down in tears because this was his life, you know. So it checked off. It checked off that the cell is novel, it's human, it's free from mycoplasma, and they also recommended that we incorporate the cell line into the global suppliers database as a cell line that other researchers can access.
Well, with a name like Henkel, sounding so much like chemical, I mean, I'm sure Henkel and is deservingly proud of his own work and in combat combination with you guys. All right, I want you to put a conclusion on this. I want you to speak to the significance of what you're doing, the relentlessness that it takes, the the moments that only you can tell that on your journey to this that will help our brothers in the Caribbean and in the wider diaspora, the significance of this, and why should I, as a viewer, understand and why this is significant? Where do you see it going? And the simple question was asked, why were you doing this research? So I suspect you have answered that question before, but here you go again.
I gave a talk at a career development day at Immaculate, and I was invited to speak as a scientist to provide perspective of the application of science, because I find that as high schoolers, and I realized when I was in high school, persons before me and subsequently, When you do the sciences, you're steered in becoming a medical doctor. And so most students, they don't envision the application of science in Jamaica in a real way. So they get close to science when you think of the United States, a Harvard, a Yale, a Cambridge. And so I was there and I spoke to them, and a lot of persons, their eyes opened up. I didn't develop the cell line then, so there was not that great of an enthusiasm as I would have expected now.
Yes.
But I taught my students earlier this year a course on cell culture. That's a course that talks about how you develop cells or maintain cells. And for the first time in my life, my level of enthusiasm when I walked into the class peaked over 100%
Wow.
because I was not here talking about theory or what someone else put in a textbook. I was here teaching a topic that I personally was involved in. And the level of motivation, desire that I saw on their faces, in their body language, that was enough to warm my heart, to give me, I mean, chills, and to make me push further for this work. That is what we're here for. I don't want to be a scientist just for being a scientist's sake. I want to be someone who can contribute to uplifting mankind in a tangible way. And the mankind that I'm here to help are my Black people. And I want to do that not in words and not in publications that just um, push me up the rank of professor or whatever, but do it so it can help somebody. So even if I don't get to professorship, but somebody's life is changed because I refuse to give up, because I refuse to listen to the persons who said, don't do it, it's not going to work, then I would have done my job here. And so we have done this. We have made the first cell line, and I believe the Lord laid it on my heart. And I thank Him so much for taking me through those days when at times you wonder, Lord, is this you? Did you put me here? And then I had to regroup again and say, no, man, the Lord put you here, and He didn't put you here to fail. He put you here to succeed. And then I have to say, if not me, then who? Someone has to do it. Who is going to do it if we don't do it? So we're here. We're here putting our mark on changing the narrative on how research has been done over the years to understand prostate cancer. We are here changing the narrative. Are we there yet? Absolutely not. Maybe I'll never see what I need to see in my lifetime in terms of a treatment that can really help. Who I don't know. But what I know is that I need to do my part and hand the baton over to my students who will do their part, and it goes on. So I don't know if that was a good conclusion.
You have you have uh, left me thirsting for more, and I trust that whenever you speak anywhere at all, you advise us, because you are now getting another invitation to come on this program once you have done your publication, and to find out what's going on, win, lose, or draw. We want to know what is going on with this, and we have seen your tireless effort and felt the passion, and we are grateful for this, and therefore we are thanking you for your service. And one would not, you know, let it go unnoticed that you are strikingly showing that, well, God is a part of what you are doing, and therefore you feel a divine intervention in your work. And I suspect when you say, if not I, then who? Then you know that someone has laid hands on you to do this. So we will lay hands on you again as soon as you see one, wherever you're going to go to talk about this. I would like to know, because you will always come up with something different whenever you speak, whatever area you speak in. And of course, when you shall have gone to your publisher, and we want to know, we want to stay lock at step with you on this one. Again, Dr. Simone Badlal McCreath, I want to thank you for your work and your effort and pass on our best wishes to your team and everyone who interface with making this a better world.
And thank you, Michael, for having me here. Thank you for giving me this platform to share so that we can inform our fellow Jamaicans that look, COVID is running, but we have some positive things that we can rest in, that we can be comforted in, that the Lord hasn't turned his back. He's here and we just need to push through.